Dear viewers, today I have decided to speak on electrocardiogram. Let us write electrocardiogram. Briefly it is called ECG. Electrocardiogram means it is the record by which we can analyze or we can see the electrical events of the heart. Electrical events of the heart and the machine which is used to record the electrical events of the heart is called electrocardiograph, and the process by which we can record the electrical activity of the heart is called electrocardiography. Now let us look at how it looks like electrical activities of the heart. Electrical activities of the heart, if we draw the diagram, then look at what I am drawing. I have drawn a line, and then what I have drawn here is wave, and then I have wave. I have recorded the different waves. I am showing you the different waves. How you can name all these waves? This is P wave, this is Q wave, this is R wave, this is S wave, and this is T wave. So again, this is P wave. The P wave stands for atrial depolarization. Atrial depolarization. Then Q wave stands for septal depolarization. R wave stands for major part of ventricular depolarization. And the T wave stands for, S wave stands for basal part of ventricular depolarization, and the T wave is ventricular repolarization. I have used different words. I think you are not understanding what is the meaning of this. Still, I will try my best to explain each and every wave. Again, I am pointing it out. This is the P wave, stands for atrial depolarization. If we look at the heart, how it looks like? This is the heart. This is the heart. And here we have the SA node. Here we have the SA node. Then we have AV node. Then we have the bundles. The bundle of his, right bundle of his, and this is the left bundle of his, and then this is the Purkinje fibers. Why I have drawn this diagram? I want to explain the current, which is electrical current, which is generated in the heart. What is the initial point of generation is special conducting tissues of the heart that I have drawn here is SA node, and then internodal pathways, and then the electrical current is moved to left atrium. This is right atrium, this is right atrium, this is left atrium, this is right ventricle, and this is left ventricle. Now, what I want to tell you in this introductory part is we can record the electrical events of the heart. I have drawn the diagram of different electrical events. One is P wave, another is Q wave, another is R wave, and S wave, and the T wave. I have written here also, P wave stands for atrial depolarization, Q wave septal depolarization, R wave is major part of depolarization, S is basal part of depolarization, and T is ventricular repolarization. Now here, this is the first part, this is the atria. This is the atria which is depolarized fast. So that is why first P wave is there, stands for atrial depolarization. This depolarization is both depolarization of right atrium as well as the depolarization of left atrium. What I want to say is electrical activity is generated in SA node, that we call it action potential. This action potential, once it is generated, this will move to the AV node, and as well as the electrical event will be transmitted to the muscles of atria, atrial muscles of the atrial muscles, whole atria will be depolarized, and that depolarization coming through this wave, we can record this. This wave is called P wave. Now there is a gap. See this gap? There is a gap. There is no wave. There is no wave in this. Actually, that is the event which is coming in the AV node. AV node. This event, actually, we are not in position to record any electrical activity which is present in the AV node. That is shown here. There is no electrical activity. There is no wave. Now electrical activity is there, but we are unable to record. Now there is another wave is Q wave. The Q wave is septal depolarization. See, this is the septal region of the ventricle. This whole ventricle, if we talk, first there will be depolarization of septal region, and that depolarization we can record here, and that is coming in the form of Q wave. And now R wave. R wave is major part of ventricular depolarization. So this is the major part of the ventricular depolarization, major part of the ventricular depolarization, and this major part is depolarized and we can record, and that is R wave. Next is S wave. That is the basal part. This is the ventricle, this is the basal part of the ventricle, this is the basal part of the ventricle, basal part of the ventricle, and this basal part is depolarized. After major part of ventricular depolarization, basal part is depolarized. Now this is called, this we can record in the form of S wave. Now there again there is a gap. There is no activity, no wave is coming here, there is a gap. Means in this I want to tell you that is the whole ventricle is depolarized. Whole ventricle once is depolarized, then we cannot see any deflection. Now coming to the T wave. T wave is ventricular repolarization. The whole ventricle is repolarized and we can record here. Now again I want to tell you about some positive wave and the negative wave. Look at this. The isoelectric line, this isoelectric line, it is on this side and it is in this side. the above this line all waves are positive wave. above this line, ये जो लाइन मैंने ड्रॉ किया है, इस लाइन को हम कहते हैं आइसो इलेक्ट्रिक लाइन. इस लाइन के ऊपर जितनी वेव है सारे पॉजिटिव वेव है, और इसके लाइन के नीचे जितनी वेव है सब नेगेटिव वेव है. इसका मतलब व्हाट आर द पॉजिटिव वेव? P wave is positive wave, R wave is positive wave, and T wave is positive wave. And what are the negative waves we can record? Q wave is negative wave and S wave is negative wave. And there are certain places where there is no deflection of the pointer. That also we will come to know why there is no deflection. So what I want to tell you is this heart is electrical events we can record, and this electrical event, this is one cardiac cycle, one beat. In this one beat we are able to record all these waves. And if we put our body with different leads, that also I will explain what are the different leads. There are three different leads. One is standard limb lead, another is augmented unipolar limb lead, and another third is chest lead. So we will have the different waves according to the different leads. जो रिकॉर्ड मैंने यहाँ पे बनाया है, that is actually coming from the standard limb lead. Standard limb lead तीन होती है, standard limb lead 1, limb lead 2 and limb lead 3. Now in this introductory part only I want to say that our heart is able to generate electrical event, and that electrical event we can record with the help of electrocardiogram, that is the machine. If we connect our body with the leads and all these leads are connected to the machine, then we can record the different waves. यहाँ पे जो मैंने डायग्राम ड्रॉ किया है, all these waves coming from the standard limb lead 1, 2, 3. So here the different waves I have recorded, and this record comes from the standard limb lead, either from 1, either from 2 or from 3. So I will explain each and everything, what is depolarization, what is repolarization, then I will explain the different waves, and then how we can calculate the duration of P waves, duration of Q wave, duration of R wave, S wave and T wave. So if we calculate and if we come to know what are the significance of this, then we will again in future can analyze, can diagnose the problem in the heart. Now I will tell you how the electrical waves are generated. Electrical waves, I told you about depolarization, repolarization, that is part of action potential, means that is part of electrical activity. So what I want to tell you is, in this I will tell you how electrical waves are generated. Electrical waves are generated and moved, and then all these waves are translated into vector. We call it cardiac vectors. Then my aim is to tell you how all vectors are translated into ECG pattern. How vectors are translated into ECG pattern. If you remember, I told you about PQRST, that is the ECG, electrocardiogram. But this comes from the electrical currents that I have already told you, depolarization, repolarization. So in this, my first intention is to tell you what is depolarization, where depolarization there, and why depolarization is there, then repolarization, and how all these are translated into cardiac vectors, and all these vectors are translated into ECG pattern. See, let us draw the diagram of myocardial cell. The heart is made up by a cell, by cell we call it myocardial cell. Myocardial cell, and then this is cell number 1, this is cell number 2, and this is cell number 3, and this is cell number 4. So the whole heart is made by the cell, we call it myocardial cell, and they are arranged. Here we have gap junction, 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 we have gap junction. Now the resting membrane potential means it is that potential which we can see when the myocardial cell is not functioning, is in a resting condition, and that potential is minus 90 millivolt. Minus 90 millivolt. Now if we draw the diagram of action potential, diagram of depolarization and repolarization, then we must know what is the resting membrane potential, and this becomes minus, this becomes negative, and it is minus 90 millivolt. Now as soon as there is a stimulation, or action potential coming from the SA node, the SA node is different. SA node comes under specialized conducting tissues of the heart, and I am talking about the myocardial cell. I am not talking about the specialized conducting tissues of the heart. I am talking about the myocardial cell. It is present in the atria, it is present in the ventricle. Now the resting membrane potential is minus 90 millivolt. Now if we stimulate, then what will happen? There will be an entry of sodium ion. There will be an entry of cations will enter here. There will be an entry of sodium ion, and this entry due to this, the potential will be changed. This will be more positive. Say it is minus 80 millivolt. This is the millivolt, and this is the time. This side is time. Resting membrane potential of the myocardial cell, when it is resting, then the resting membrane potential becomes minus 90 millivolt. Now as soon as there is a stimulation, there will be an entry of sodium ion. Positive ions will enter into the cell, and this negativity will be turned towards positive, means minus 90 millivolt can be minus 80 millivolt, or this can be minus 70 millivolt. Now this minus 70 millivolt, we may call it, this is minus 70 millivolt, we may call it threshold potential. Once it is threshold potential, once this potential is reached up to threshold potential, due to the entry of sodium ion, cations enter into the cell, then there will be a stimulation of voltage gated sodium channel. The voltage gated sodium channel, due to this potential will be changed, and this minus 70 will be turned to plus 40 millivolt. Now what will happen? In this the polarity of the cell is changed, is altered, and this become negative is changed to plus 40, or this is polarization of the cell is gone, is reduced, is gone. So that is why this is called depolarization. Depolarization. Polarization is absent, is turned to zero, and then maybe 10 millivolt or 20 millivolt or 30 millivolt or maximum 40 millivolt. And this polarity is changed here. That is why this is called depolarization. Then there will be a stimulation of calcium channel, and there will be a stimulation of potassium channel. So the exit of potassium will be there, as well as entry of calcium will be there. So what will happen? Because of this, see here, this potential is again changed. The potential is again changed. Changed means, what I want to say is, this plus 40 now is again going towards negative. Means that, and that come from the interaction coming from the both calcium channel and the potassium channel. Calcium channel, calcium ion is coming in, and the potassium channel is going out. So as you all know, if potassium is going out, means the cell will be again moved towards negative, and the calcium ion is cation. This will try, means this channel will try to not to make the cell more negative. So there will be a continuous interaction between potassium ion and calcium channel, potassium channel and the calcium channel. Now ultimate result is, we will have the plateau phase. Means positivity of the cell will be maintained for some time, and then the calcium channel, function of the calcium channel will be lost, the potassium channel will be more active, and this potential will be again coming back towards again minus 90 millivolt. So this is called repolarization. This is called repolarization. If we look at the time here, this takes near about, this is 100 millisecond, 200 millisecond and 300 millisecond. The time is given here in millisecond. Means the whole duration of action potential, if we look at, it becomes 300 millisecond. In this we have depolarization, and then we have repolarization. That I am talking about one cell. But what is happening with the second cell, neighbor cell, third cell and fourth cell? Due to this gap junction, once there is an entry of sodium ion, again look at, once there is an entry of sodium ion, the sodium ion enter here, sodium ion enter here, sodium ion enter here. So what is happening? The graph which we are looking here, the same type of graph also we can see here. The graph which we have recorded here, the same type of graph we can see here also. Means here also it is millisecond, here it is millivolt, here it is millivolt, and here it is millisecond. So total duration 100, 200, 300 millisecond. This is in millisecond, 100, 200, 300 millisecond. Here we have depolarization, here we have repolarization, here we have plateau phase, here we have plateau phase. So what I want to tell you is, as soon as there is stimulation of single cell, there is a stimulation of again rest of the cells also. So because what I want to tell you is, there is a gap junction. So whatever changes we can see here in cell number 1, the same type of changes we can see in cell number 2, and the same type of changes also we can see in cell number 3. So what I can say is, once the action potential is generated here in one cell, this wave is generated and it is transmitted from one side to another side. So this is the action potential. Means I am talking about depolarization and repolarization. I am talking about electrical waves. Once it is generated, it is moved. Here, say this is the heart. This is the atrium. This is the right atrium. This is the left atrium. This is the right ventricle, and this is the left ventricle. So we have SA node. Here we have SA node, and this node actually comes under specialized conducting tissues of the heart. This node is responsible to generate impulse by its own. Once it is generated, then the role of this is electrical impulse, electrical current in the form by action potential. Once it is generated, it has to be moved. This will stimulate the myocardial cell. Once myocardial cell is stimulated, then as we already have seen, cell number 1, cell number 2, cell number 3. So the all cells present here will be stimulated. Look at this diagram here. This is row number 1. This is cells, we can see here. This is row number 2. This is row number 3. This is 1, this is 2, this is 3. The electrical action potential is generated here, and then it is moved towards this, moved towards this, moved towards this, towards this, towards this. Then again it is moved towards this, towards this, towards this. So this way, this electrical current in the form of action potential is moved from one side to another side. This small arrow, this arrow indicates the direction of electrical current. To which direction this current is moved. This current may be moved towards this. This current may be moved towards this, towards this, towards this, towards this. That is the direction. And this direction of the electrical movement, this direction may be small or maybe larger one. This direction of electrical activity, electrical movement, we call it vector. The direction is vector. And again, the direction, if you draw it like this, this is the vector, and this vector may be positive or this vector may be negative. That also I will explain why we call it positive vector, why call it negative vector. This is the positive vector, this can be positive vector or this can be negative vector.